Hello, my dear students. Now, this is my 13th lecture, week 3rd, in the protein quantification. Myself, Mehdi Hayat Shahi, Assistant Professor, Interdisciplinary Brain Research Center, Faculty of Medicine, Aligarh Muslim University, Aligarh, Uttar Pradesh. The content of my today lecture is introductions about protein quantifications. Then we discussed different assays, and then we have discussed BCA assay, Bradford assay, and Lorry assay, then UV spectrophotometric, and then we discussed Haldol methods and also fluorescence based assay. The references I have taken for this lecture from Lubertus Trier Biochemistry, 8th editions, then Harper Illustrated Biochemistry, and The Principle of Biochemistry by Albert Lellinger. Introductions. Protein bioassay, the protein quantification. It is very essential step in the process of protein isolysis and also for analysis of the proteins, because we need to understand downstream assay. Means we need to quantify protein. That's why we need to know by the downstream assay, like for Western blot, ELISA, or any enzymatic assay. That's why we need to quantify the protein. And it is ideal, it should be rapid, means times, and also it should be sensitive, reproducible, and resistant to interferences. So these are the ideal assay characteristics we need to take care when we quantify the protein. Uh, the assays, the BCA, biocyanoconic acid, biocyanoconic acid assay, also called Smith assay. It is widely used for total protein quantification. It's very common in the lab. People use also different condition we use for this, very common. And it is suitable for detergent containing samples. If samples have detergent, associated with detergent application, or buffer has detergent, then it's good for that. And also it is good for the complex biological acids. Complex biological acids. Then another assay, this is a Bradford assay developed by Bradford in 1976, and it is based on Kumasi beryllium blue dye, and it is very fast and also is very simple method. BCA, the principles of BCA, the protein reduces copper 2 plus to copper 1. That's called Biuret reactions. So 1. And what happened? BCA chelate Q1 copper 1 purple complex, and then we have to measure at 595 nanometer. And this is absorbance versus protein concentration. So whatever we got the observation, we have the protein concentration. Higher the observation, we higher the protein concentrations. And the reagent and the materials we use for this, BCA solutions, copper sulfate 4%. Then also we use BCA woman serum albumin standard, that is 1 milligram per ml. And the materials which we required for BCA assay, that is a spectrophotometer and macroplate reader, that's also called ELISA reader. Then we need pipettes and also test tube and cuvettes we need for this. How it works? We got the two reagents, and that is reagent A and reagent B. Reagent A and reagent B. First, we mix reagent A and reagent B in 50 to 1 ratio. That is the ratio. The reagent A is 50 and reagent B is 1. For example, if we take 5 ml of reagent A, then we are going to add reagent B 0.1 ml, and we mix well. And always whenever we perform any protein quantification, we have to be freshly prepare the samples. Then after this preparation of working reagent, we perform or we prepare a standard which provided by this BSA kit. In this case, we prepare BCA standard. Example is here. First we have 0 concentration, then we have 125 macrogram per ml, then we 250, then 500, then we have 750, and we have 1000 macrogram per ml. And this is the standard, that's called S0, S1, S2, S3, S4, S5. This is a standard of BCA. And we use the same buffer as a samples, same buffer as a samples. The same buffer is the samples in this case. Now we have a ELISA plate. This is the ELISA plate, 96 well plate, 96 well plate. And what we do in this case, add the samples and a standard. In this case, we add 25 macroliter of each standard and sample into wells, in this wells. But it should be in triplicate because it's important. We use in the triplicates. After adding samples and a standard, now we add working reagent which already we made a solution. In this working reagent, we add 200 macroliter of working reagent in each wells. Then mix in the shaker. We use shaker to mix the plates, this 96 well plates, and also is important to avoid the bubbles. Then after mixing, we incubate this 6 well plates, and also we need to protect from the strong light. So we cover this plate with aluminum foil and incubate at 37 degree. We normally incubate in the incubator for 30 minutes, or we use, if wanted to incubate for the room temperature, so we need to use 2 hours incubations. And now we need to measure the absorbance after 2 hours or 30 minutes depending on the temperature, and read the absorbance at 560 nanometer. This is the ELISA reader. Or now we calculate the protein concentration. Calculate the protein concentration. What we do in this case, we draw a plot, a standard curve based on the absorbance and also the concentration of BCA. This is the x-axis. We have BCA concentration in macromolar per ml, and this is the y-axis. We have absorbance in 560 nanometer, and we got a straight graph, straight line graph for the standard. This is 1000. This is 175 and 500, and this is 200. So this is a standard graph we got, and then finally we have the samples. So we can have samples something like here or something like here. So on this basis, we can calculate the concentration of a protein in the samples. Important notes or important point we need to consider when we do BCA assay. It is compatible with SDS, sodium disulfate, although in not very high, low concentration. Also with Triton X100, NP40 and REPA buffer. But it is interfere with beta mercaptolithanol, DDT, and also a strong chelator like high EDT concentrations. And also we take consideration when we run, always include blank. In the case of blank in the buffer, there is a buffer, and we have to run a standard also sample in triplicate. And if we have observations is too high, like more than 1.5, then we have to need to dilute the samples. And also we keep all readings within the linear range, in the linear range. This is the tips, or the condition we can keep in mind when we perform BCA assay. Now the absorbance curve and interpretations. Higher absorbance has higher protein, lower absorbance has lower protein. This is a criteria. The formula when we get the absorbance, the percentage of absorbance is equal to A sample minus A blank divided by A control minus A blank multiplied by 100. So this is the criteria, we can have percentage of absorbance to calculate. Now the Bradford assay, in this case, dye bind to the different protein specific, that is arginine, lysine, histidine residue, and color change from brown to blue. This is the characteristics. And also we measured the concentration or reading we take at 595 nanometers. In this case also we prepare a standard curve and calculate. We prepare the calibration curve, and this plot is also absorbance versus concentrations, and we determine unknown using linear equations. Determine unknown linear, we use based on the linear equations. And how can we interpret absorbance? High absorbance means more protein. Low absorbance means less protein. What are the materials and reagent we use for this case? We use BCA standard from 0 to 50 macrogram per mole, and also Bradford reagent we use from different company, in this case Bired, when we use deionized water, and we use macroplate and the cubate. Now the Bradford assay, prepare Bradford reagent. So the Bradford reagent is Kumasi Brilliant Blue G250, and we dissolve this in the phosphoric acid and make a solution. Then we have a sample standard. First we have a blank, and the concentration is, in this case, this is only the buffer 1 ml, we know concentration, the reagent. Then we have 5 milliliter standard 1, then we have standard 2 is a 10, then we have a 15, 3, then we have a standard 4 is 20, then we have a standard 5 is 25 milliliter, then a sample, this is known, we have to calculate, that is X value. Then we mix the samples, vortex. Well, then we incubate 5 to 10 minutes at room temperature. Again, we measure at length, wavelength 595 nanometer. Then again, we generate the standard curve. Y-axis is the absorbance and x-axis is the protein concentration. And absorbance is 595, and we got a straight curve. This is a standard. Now we measure the sample absorbance, and sample absorbance at 595 fall between this concentration. So we, based on this concentration, we can calculate the protein concentrations. To read the concentration from the standard curve, and we calculate the protein concentration in macrogram per mole of the proteins. What the principles and apply in the Bradford assay? Comasi Brilliance Blue G250 bind to the protein under acidic condition. In this case, we use acidic condition, and causing a shift in the absorbance in maximum at 595 nanometer. In this case also we use BCA as a standard, and also in this case we ensure sample are free from interfering substance like detergent and buffer. So that's a limitation for Bradford assay, we to avoid detergent and buffer and salt. And also we got a linear in the range of 1 to 1000 macrogram per ml, and it is applicable the protein quantification for SDS page and enzyme assay, and also for cell lysate. This is the criteria for Bradford assay. Now we compare the Bradford and BCA assay. The principle for the Bradford is a dye binding. Comasi binds to the protein, but for BCA it is a copper 2 plus 2 copper 2 is a reduction process. In BCA lost 1 proton, this is reduction conditions. Then color change in the case of Bradford is a brown to blue, but in the case of BCA green to purple. Also they have different wavelength. In the wavelength in Bradford is 595 nanometer, in the BCA is 560 nanometer. The sensitivity of Bradford is moderate, but BCA is very high. A linear range, Bradford has narrow, but BCA has wide range. and time is required for Bradford is hardly 5 to 10 minutes, but BCA required 30 to 60 minutes. And also the protein viability in the case of Bradford is very high, depend on amino acid, but in the case of BCA is low, more consistent. And for the detergent compatibility, Bradford is very poor because SDS is interfere in the assay. But in the case of BCA is very good, it handled SDS and REPA buffer. Reagent, reducing agent, we use beta mercaptolone and DTD is very compatibility in Bradford assay, but it interfere in the BCA assay. And reproducibility is very moderate in the broad, but is very high in BCA assay. Now the Lorry assay also one of the method to quantify the proteins. In this case also we prepared the standard samples, that is BCA standard in the concentration macrogram per mole per ml, and that is 0 to of 100. This is the samples, and we keep the sample in the ice. Then we add reagent A, that is alkaline cupric solution, alkaline cupric solutions. So we have to add 1 ml of reagent A in each tube and mix well. Then we have to incubate for 10 minutes at room temperature. In this case, we need room temperature. What happened? Incubate at room temperature for 10 minutes. Then protein plus copper plus 2 ion become oxidation further under alkaline condition, and the loss one hydrogen ion, then the Q plus. Then after this 10 minutes, we add reagent B, that is called Folin Chico-Chelatu reagent. This we add 0.1 ml or 100 microliter of reagent 2 in each tube and mix immediately. After adding, we have to mix immediately. Now we further incubate for 30 minutes at room temperature, and then this blue color appear. In this case, the blue color appear. And now we have to measure the blue color. And this case, the wavelength is 750, the good absorption or maximum absorption we have, measure absorption 350 against reagent blank. We have to compare with the blank. And we use in this case a spectrophotometry. This is a cuvette we use for measurement to add the solution in the cuvette. Now further, we prepare a standard curve and determine the concentration. On the again the y-axis we have absorbance maximum at 750 nanometer, and the BCA concentration in this case 0 to 1000 in macrogram per ml, and we plot absorbance and versus BCA concentration, and then we determine the concentration of known sample from the standard curve. This is the method as BCA and Bradford. And the principles of this Lorry assay, that this is a protein and then this is a reagent A. Then we have a reduction, the loss 1 hydrogen, the copper, 1 hydrogen atom. Then this reagent B, when add, this become blue color complex, and we measure the absorbance. Then we also have UV spectrophotometric method, that is also called A2T method, A280 methods. The principle of this method, protein absorb at UV light and 280 nanometer. This is responsible for due to two amino acid, that is tryptophan and tyrosine. And it is advantage for this, it's very fast, no reagent is required, and it also non-destructive when we can use even nano drop, but it is very low accuracy in the case of impure proteins and also nucleic acid contamination. That is a limitation for the UV spectrophotometry methods for the protein measurement quantifications. This is the photospectrophotometry. We measure the protein quantification. We do the quantification to measure protein concentrations. Now we have Kajadal methods, that is classical methods. It measured total nitrogen content. Based on that principle is total nitrogen content, and what this method does, is convert nitrogen to the ammonia and is going to quantify. It has advantage and also it has some limitations. It is accurate for total protein content and which we use in the food and industrial analysis. But it is time consuming and it is non-specific, non-protein nitrogen also included in this method. And what is the flow chart of this method? First, we take the samples, then we have to digest the samples. Then we perform distillations by helping of neutralization and dilution of the sample buffer or solutions. Then we perform titration and then we go for the calculations. So this is a flow chart for cardinal methods. It's also called classical methods. What apparatus we need? We need this Cadazal flask, digestion block, distillation unit, burette, chronicle flask and pipettes. The reagents we use concentrated hydrogen sulfate catalyst mixture, then sodium hydroxide, boric acid solution 4%, then we need a standard hydrochloric acid 0.4, and then also we need mixed incubator. And this is the overall chemical reactions. This is organic, and then we have hydrogen sulfate catalyst, then we have form ammonium sulfate, then ammonium sulfate, hydrogen oxide, sodium hydroxide form the ammonia release, then nitrogen sulfate, then water release to water molecules, then this further ammonia react with boric acid solutions, then we have ammonia and then H2BO3 in solutions. Then finally this H2BO3 get hydrogen bond and become H3BO2 at the end. This is the reaction process, overall chemical reaction of Kajidal methods for quantification of the protein. Now we have fluorescence based assay. It's very high sensitivity. In this case, we use fluorescence dyes bind to the protein and emit signal. The advantage is very high sensitivity, even nanogram of protein we can quantify. And also it has less interferences, but the limitation is this is very expensive and also it's require fluorescence reader that is expensive. Now the summary of my lecture, different methods we use. BCA sensitivity is very high, speed is medium, and interference is low, and it is used in journal lab. It's very common BCA assay for protein quantifications. Then we have Bradford, sensitivity is medium but is very fast, and interferences is moderate and is very quick screening. Within 10 minutes we can get the protein concentrations. Lorry is very high sensitivity but speed is slow, and interference is also high, and it's in the research accuracy, it gives good quantification or accurate quantifications. UV 280 nanometers when it has low or medium sensitivity, but its speed is very fast, very quick, we can do quantification, but interference is also very high. And for this purpose, the best use we can use pure protein, because in the impure protein there is interferences by the different like nucleic acids or some salt and solution buffer. Then the fluorescence is very high method, but speed is medium and interference is low, and also we need like best use we can have low concentration of protein we quantify with the help of fluorescence method. Then we have Jethal method, that is very high sensitivity and speed is very slow and interference is also low, and it is applicable for the food and industrial purpose, not for general lab use, because it converts, but that also it has limitation because it convert the nitrogen, all nitrogen, non-protein nitrogen also in ammonia. That's the limitation. Thank you so much for listening this lecture.